I'm Wendy Hartsock, science and peptide enthusiast. In this episode of Exploration Science, Professor David Craik from the University of Queensland talks about discovering the cyclotides, as well as their potential to transform medicine and agriculture. I was going back through, your publications, obviously, but even through the American Peptide Society meetings. And really, I was trying to think back of when I first started hearing you give lectures. And I think the first one was 2007, when you talked about, I think it was sunflower trypsin.
Sunflower trypsin, yeah. So making me feel old now.
Well, I'm sorry.
That's because I am old, but yeah. Look, to be honest, Wendy, I've been to so many conferences, I can't remember which conference I talked at, where they all sort of blur into something.
Yeah.
And when people point them out, I can remember, oh yeah, that was in Boston. But where was that one? That wasn't Boston.
That one was in Montreal.
Oh, okay, yeah.
Bill LaBelle's conference.
Yeah.
And that was my first APS, and then I was hooked after that, so yeah.
Well, I hadn't been in peptides for too long. That may have even been my first one. I started doing non-peptide stuff. I'm an NMR person basically, so I sort of got into peptides years after.
Yeah, how was that? How did that happen? Because I did see that the early papers were on NMR.
Yeah, I did a sabbatical over at Oxford, and I wanted to learn how to do NMR structures. So I was doing NMR of small molecules, antidepressant drugs and thyroid hormones and things like that. And I wanted to learn how to do structures of peptides and proteins. So I went to Ian Campbell's lab at Oxford, which was the place to go. It seemed to be a magnet for Australians and people from the Commonwealth to go and learn about NMR. So I went there. I only had 4 months. And I just said I'll fit in with whatever projects you've got going here. And they didn't have any small projects, except there was this weird peptide that had been sitting there for years that a Norwegian postdoc had left earlier. And it turned out to be the Kalata molecule. And they hadn't been able to do a structure at that stage. So it was my job. They said, do you want to look at this? Because no one else wants to look at it. So I ended up doing the structure of it. And that's when we discovered the cyclic backbone and the cysteine knot. So then I got hooked into peptides after that.
Right. So was the Kalata B, that was, I guess, so you found that first and then you went looking for it in nature. So can you tell me the story of how that?
Yeah. So the story was that a Norwegian doctor had gone down into the Congo in Africa in the 1960s. And when the Congo became independent of Belgium, then all the local tribes wanted to work out who was going to be the boss. So there was lots of tribal wars, and the Red Cross sent people down there to try and fix up the injuries. And a Norwegian doctor, Lawrence Brand, went down there. He was actually an anaesthetist who was the Red Cross sent lots of surgeons, but they forgot about people that could deliver anaesthetics. So he actually got called back from his honeymoon to go down and deliver anaesthetics. And his first patient had a spear going in through his mouth and out the side of the cheek, and he had trouble working out how to get the anaesthetic mask on. Anyway, he went down to serve in the Red Cross, and while he was there, we just noticed in the local hospitals when women went into labour, the birth just was over like this, and the women were very secretive about why it was happening. And it turned out that they were making a tea from a local plant called Oldenlandia, and that was accelerating the childbirth. And he took some of the plant back to Norway and discovered that the active ingredient was a peptide, which he called Kalata fraction 1, so flat Kalata B1. And so he discovered this uterotonic peptide in a natural medicine that had been used for decades in Africa. But this is in the early 70s and couldn't really determine the structure. Knew it was stable because the women were boiling it to make the tea and then ingesting it. And, as you know, most peptides wouldn't survive ingestion, and we still don't have an insulin tablet after 100 years of use of insulin because it would be chewed up by proteases. But this peptide was super stable. And then nothing happened. He wrote a PhD up on it. Nothing happened for 20 years until I stumbled across a sample of it because another Norwegian postdoc had gone to Oxford. They gave it to me as a little project, and it was my first NMR structure. I've got my little model here of the actual structure of the Kalata molecule. So it's a circular backbone of 30 amino acids with 3 disulfide bonds, and that actually hadn't been seen before in literature. And I just thought I've done something wrong. This is my first attempt to do an NMR structure. I couldn't believe you could have a disulfide bond threading through a ring of 2 other disulfide bonds. I didn't actually publish it for 3 years after we had the structure. And no one had seen a peptide of that size with a cyclic backbone either. So I would go to conferences and talk about it, and people would say, "You're crazy,"
Okay, yeah.
possible. But it turns out to be true. And so, I thought if there's one example, maybe there's other examples.
Yeah.
So that was my excuse to travel around the world and dig up plants from all sorts of places. I started in my backyard in Brisbane and then gradually branched out and got to go to more and more exotic places of course, you always have to go to an exotic place to find the most, different plants. And it turns out that they're quite, it's quite a wide range of plants that make these molecules.
Right. And so, yeah, maybe you talk about the plant diversity, and then how many cyclotides have you and others discovered?
Right now, we've got a database, and there's somewhere between 600 and 800 actual sequences reported in that database, which we call Sybase. But we've predicted that there's easily more than 50,000, probably hundreds of thousands of these things, just based on the rate of discovery. And now, of course, we're doing a lot of work at transcriptomics and genomics level. So I think it's going to be a much larger family than the actual ones we've sequenced so far at the peptide level. Many more gene sequences to come over the next couple of years, I think. So.
Right. And then within the plant, what is the diversity that you see across?
Yeah, so within the plant kingdom, we find cyclotides in 5 major plant families. So originally they were discovered in the Rubiaceae, so the African plant from which it all came. Aldenlander affinis is a member of the Rubiaceae. The Rubiaceae actually is also colloquially known as the coffee family. So coffee plants actually come from that family. And in fact, that was one of the first plants I looked at when I saw it was from a Rubiaceae. I've actually got 2 coffee plants in my backyard, so I extracted from the coffee plants, but unfortunately no cyclotides. So I can't say that they've been in human, clinical trials every morning. But the Rubiaceae was the main one. Then we looked in the Violaceae, or the violet family, and there's about 900 species of those around the world. The Cucurbitaceae, or the cucumber or gourd family. The Fabaceae, which is sort of legumes and butterfly pea. And the Solanaceae. So Solanaceae is the family that contains tomatoes and potatoes and petunias and those sorts of things. So we don't find them in actual tomato plants, although we are putting the genes into tomato plants. We don't find them in potatoes, but we can transform potatoes. But we do find them in petunia plants. So a lot of your listeners have probably got petunias growing in their backyard, so they've certainly got cyclotides in those. And certainly if they've got violets or pansy plants, then there'll be lots of cyclotides in those.
And why
in those 5 families?
Okay. And why are plants growing these and making cyclotides?
Well, we think that they're naturally produced as defense agents. So I always like to say, if you're an animal and you're attacked by a predator, then typically what you'll do is run or fight or scream or scratch or bite. But plants can't do any of those things. So they've evolved lots of chemical defenses. And, in the past, we've looked at lots of those molecules, like caffeine, nicotine, things like that are basically chemical defenses for plants, which we use for other purposes. But over the last 10, 20 years, I guess people have realised that plants actually produce these wonderful peptides, including cyclotides. So cyclotides are basically there as natural insecticides, building up in the leaves of plants, ready for when a caterpillar comes along, chews on them, then the cyclotides will have their insecticidal effects.
Okay, so then it's interesting that the Colada bee was utilized for, I guess, analgesia associated with pregnancy or with labor pain, is what it sounds like. So,
not so much labour pain, but more uterotonic, acting on the uterus to accelerate contractions. Yeah, so it's a completely. I mean, there's no reason why a plant would make a uterotonic agent.
Yeah.
Although there's sort of anecdotal reports. People say in Africa, elephants maybe start to eat some different plants when food supply is short, and maybe there might be some natural. The elephants have discovered that if they have these plants, then they can make the birth easier or faster. Right. I don't know. That's anecdotal stories we hear.
Huh. Interesting. And then I guess along those lines, there are other areas that you've looked at for cyclotides. So maybe you could talk about like the different therapeutic areas or disease states that you're interested in.
Yeah, so when we first discovered them, we didn't know what they did. We just knew that they were weird molecules that were super stable and that one of them had this uterotonic effect. And then we thought that their natural function is insecticides, and we pursued that work, which I can talk about later on. But in the meantime, I thought, well, if this peptide can be boiled and orally ingested, it's super stable, and we learned that from the unusual structure of the cysteine knot and the cyclic backbone. And we thought, well, the world doesn't really need another uterotonic agent. Oxytocin works just fine. But the world does need to stabilize peptide drugs. And if we could take this, this stable peptide framework from the cyclotide and maybe graft on an epitope onto one part of it, we could introduce a new functionality. So that's what we call grafting. And it's a bit like, if you're a gardener, plant grafting, where you take a rootstock and graft something else on. With cyclotides, we take the stable framework and then graft on a peptide epitope that either we've discovered in our lab or a pharma company has discovered, or we use phage display. And what we hope is that when we graft that peptide epitope, it'll get the stability property of the cyclotide framework and retain its activity. And by and large, that's been quite successful in the lab scale of working. So there's one cyclotide molecule that's in clinical trials in Sweden at the moment for multiple sclerosis. So this is work that sort of is being put forward by one of my ex-PhD students, Christian Gruber, who's now in Vienna, and that company's taken it through phase 1 clinical trials. So we're very excited to, that would potentially be the first cyclotide to make it to humans if it goes through.
Sure. So what is the biodistribution of that cyclotide?
That cyclotide is orally active, and this is one of the reasons we were excited about cyclotides in the first place, that if the women are making the tea and ingesting it, then there's obviously oral activity. So we were very excited in the early days that we cracked the holy grail of peptide based oral bioavailability. Unfortunately, the actual oral bioavailability is not high. It's less than 1%, as is typical of most peptides. But the fact is, because some of these molecules are so potent, you can actually have oral activity without having a huge oral bioavailability. So there's still challenges. We still need to get the actual oral bioavailability up. The pharma industry really wants, at least 20% oral bioavailability to, avoid intrapatient variations and things like that. But so, we can't claim that cyclotides are 20 or 50% orally bioavailable, but some of them are certainly orally active.
Sure.
And we've got to, work on bumping up the oral bioavailability. So that molecule for multiple sclerosis was originally delivered orally, and the assays in animals showed oral activity. And we've got a number of examples where we see oral activity of these cyclopeptides.
Right. Okay. So, sorry, I have like 20 questions I want to ask at once.
Yeah,
I gotta pick one story to go down. I guess something that, maybe you could talk a little bit, tell me about the ease of synthesis of these molecules, but then where you really, where you want to take them, how do you see them evolving as you make medicines out of them?
Yeah. So cyclotides are obviously naturally produced by plants, and plants produce them very efficiently. So plants that produce cyclotides, 1 gram of the weight for every kilo of plant material, there's 1 gram of cyclotides. So that's a huge amount. It's more than the small subunit of rubisco, photosynthetic protein. So plants are very efficient at making them, and they're made, they're genetically encoded as precursors and then cyclized. So as chemists, in the plant cell, they're made by, first of all, expressing the linear peptide and then cyclizing later. As chemists, we do the opposite. We use native chemical ligation. So we've borrowed the wonderful work of Phil Dawson and Tom Muir and Steve Kent and others to form a cyclic backbone first, and then we form the cysteine knot in the subsequent step. And that seems to work quite well. People say, how can you form that complex cysteine knot? But we don't do anything particularly clever to do that. It seems as though that's a thermodynamically favoured structure. The one little trick we do in the folding buffer is often we add a little bit of isopropanol. The interesting thing about cyclotides is that as they fold, they become more hydrophobic, I guess, because the hydrophobic amino acids that normally pack into the core of a protein there's no room for them because that's full of cysteines, the cysteine knot, which is hydrophobic in itself. But some of the other hydrophobic residues get forced to the surface. So a correctly folded cyclotide moves to a much longer retention time on HPLC. So they're really easy to purify. You just look for the single peak by itself standing out from the crowd. For most cases, that's relatively straightforward. So not too complicated.
And I've seen that in other peptides that have multiple disulfides, that it's almost like one snaps in and then it kind of guides the rest to snap into place. Yeah, very interesting though. So, but do you see kind of, because I know, you talk a lot about making making medicines in plants, because that obviously can assist in distribution, and there's a lot of benefits to that. But do you see these sort of going, I guess the one that's in clinical trials for MS, is that produced synthetically? Or is that?
At the moment, as I said, I'm not directly involved with that company, but I know that they are producing that synthetically, or contracted to produce that synthetically, which is the way that things would normally go in the early stages of development. And so, I don't think we're going to replace peptide chemists by plants as factories. Good luck. We all need the jobs going forward. But I'm really excited about other possible routes of synthesis of cyclotides. We know plants can do it wonderfully well. We know chemists can do it wonderfully well. I should say there's one class of cyclotides we can't really fold well. There's three classes: the Moebius ones, the bracelet ones, and the trypsin inhibitors. And the trypsin inhibitors and the Moebius ones we can fold beautifully. For some reason, we can't fold bracelets. They're not that different from Moebius ones, but we just haven't cracked that yet. Yet those are the most common ones in nature. 2/3 of all cyclotides are in that bracelet subfamily. So obviously plants have got the right protein disulfide isomerases or other enzymes to do the cyclization. So we haven't yet cracked that as chemists.
Right.
But one of the things that really excites me is this idea of maybe using plants as biofactories, because, as I said, plants produce the cyclotides in huge amounts. If we can engineer a cyclotide, design a cyclotide that we can put into a plant, then in theory we could get peptide-based medicines out into developing countries where we can get the people growing medicines, maybe not in their backyards, but under some sort of controlled circumstance, or maybe even in their backyards for certain medicines. So with the multiple sclerosis example at the moment, we're actually toying with the idea of manufacturing that in tomato plants. So I've got my little tomato plant here as a model of the sort of thing that we can do. So imagine, a person with multiple sclerosis, that this is not a cure for multiple sclerosis, it's a lifelong condition, but it could reduce the symptoms. So, you could be having your Greek salad with the tomatoes in it that would sort of minimize the symptoms going forward, or your tomato juice in America, tomato juice over here. And my favorite would be to put it in a Bloody Mary, so that you can sort of have the best of all worlds, and so have your medicine. So, I mean, I'm sort of sounding as though this is trivial, but in fact it's not. There's lots of cases where people do need to take medicines lifelong, and if we can think about selected cases where that could be in food, then I think there's ways of doing that now technically.
Yeah. So this is, I'm not being facetious yet, maybe a little bit, but you brought up Bloody Mary, and I know we've talked in the past about beer. It's definitely a connection that I feel with you with the gluten-free beers. But I guess alcohol itself does actually improve, or it can be good or bad, I guess how you look at it, permeation of the gut. So actually there might be cases where that could help with the bioavailability. Have you studied that at all?
No, we haven't actually studied that, but I agree with you entirely that it could be that the formulation could be really important to delivering some of these medicines orally. And I'm not recommending that people drink Bloody Marys every day as their therapeutic. I should say that there is a gin out, at least in Australia, called ink gin. That's a blue-coloured gin that is made from the colouring comes from the butterfly pea flower, which is a beautiful blue flower. And that changes colour as you add your tonic water, and the pH changes. It's actually an anthracyanin that has the chromophore change. It's been around for a couple of years in Australia now. I think it's gone internationally, but we know that flower, butterfly pea, contains cyclotides. So we actually ran it through the mass spec on one occasion, wondering whether cyclotides would survive. And in fact, there's about a dozen cyclotides in the commercial gin. sitting there that are detected on multi mass spec, showing that actually cyclotides can sit in 43% alcohol, survive storage at several months in the liquor store, and be ingested by humans and not have harmful effects. So I think you made a good point that the fact that cyclotides can exist in these alcohol mixtures shows that there's some potential for formulation. And of course, we know that cyclosporin, which is probably the best example of an orally bioavailable peptide, that's heavily formulated with olive oils and all sorts of other things to help the bioavailability. So the fact that cyclotides are so stable means that they are actually amenable to a whole range of formulation type tricks.
So the bioavailability of cyclosporin isn't that high either, right? I mean, it's
no. I think it gets up to about 27% in a formulated type product. I think natively it's probably lower than that.
Yeah, definitely. So when did people start to kind of come around with the cyclotides? Like how much data did you have to have before people were like, oh yeah, Craik's not crazy?
Not completely crazy. Well, I still think I'm completely crazy. But look, when we started this, we actually did the structure of Colada B1 in 1991.
Oh, wow.
And we didn't publish it till 1995 because I wasn't 100% sure that, we hadn't seen a cysteine. No one had seen a cysteine knot when we first did the structure. Some X-ray structures of cysteine knots started to come out in the 93s, 94s, and that's when we sort of thought, well, this could be true. But because we'd done the structure by NMR, in those days, it was very early days of NMR structures of peptides and proteins, so I wanted to be sure. So I guess 95 is when we published the first structure, and no one, I don't think people completely believed it at the time, but by 1999, I guess we discovered dozens of other examples, and there were a few other groups that were working on them. There was a group in Sweden, a group at the National Cancer Institute in the US, a group at Merck that had all found similar molecules. And so I guess, when it's coming from multiple labs, people believe. And then we started, once we developed the chemistry to make them in the early 2000s, I think they become a bit more accessible. So I think it's a semi-respectable field at the moment. So we're pushing on with it.
Well, and so then as you're discovering more, I guess, you mentioned that some of the most diversity comes from exotic places. Where so far has been your favorite adventure in discovering a cyclotide?
We do a lot of work in Southeast Asia, and so I travel to Thailand quite a lot. And I've got a good friend who's a botanist that knows a mountainous region between the Thai-Cambodian border. It's not a very high mountain, maybe 2,000 meters, but it's a very inaccessible place that not many people have gone to. And he lived in that region for years, and every day would hike and mark some of the trails. And so I got to go on a couple of wonderful field trips to the top of this mountain in a place called Khao Soi Dao. That, there's probably only maybe a few dozen people in the world that had ever been to this particular place, but he knew that there was a particular type of violet there. So we got to go there, but that involved carrying machetes literally to chop our way through the jungle, carry your tent and food and water for a couple of days. So that was probably the most adventuresome and good in terms of being able to find a violet that is not present really in any other place in the world, and find that it contains cyclotides. So that was a memorable one. But we've been to China, into South America, India, or Sweden, all around Hawaii. There's some really interesting violets in Hawaii that we're very interested in tracing the evolution of violets across the Hawaiian Islands and see if we can track the evolution of cyclotides within the different species of violets on Hawaii. So that's my excuse to go to Hawaii. Every now and then.
Evolution.
There's one wonderful place on Kauai in the Alakai National Park, which is, I think, allegedly officially the wettest place on Earth. It's always drizzling with rain there, and there's a swamp at the top of that, and there's a couple of species of violets there that are really interesting as well. That have got some interesting cyclotides in them.
Right. So maybe you mentioned the insecticidal properties of the cyclotides, and I believe you've presented before on what some of these are doing to insects when they ingest them. Are those the natural cyclotides, or were those cyclotides that you had grafted additional insecticidal properties onto?
No, actually, those are the natural cyclotides.
Okay.
So, and they come from the plant called butterfly pea, Clitoria ternatia. And this is work that was actually done by a local Australian company called Innovate Ag that had discovered that this plant, when it was in near cotton fields, that it wasn't attacked by insects, and the cotton that was nearby wasn't attacked by insects. So they actually made extracts of this plant, so all natural extracts, and found that if you sprayed that extract onto other plants, like cotton, for example, that it would provide some protection from insects. But they didn't know how it worked. And then independently, we'd looked at that plant and found that it contained multiple cyclotides. And in fact, we did the transcriptome and find that there's 70 plus cyclotides in it. So we're able to work with that company and help them with the registration process and understand how this product works, which is, we're not allowed to do ads on your show, but I'll just show you the product.
There's no rules. So it's called Cero.
X. So the reason the company called it that, Cero is Latin for plant, so they called it Plant X. They didn't want competitors to know what the plant was, but they can tell you now it's butterfly pea. So this is this wonderful plant that produces at least 70 cyclotides that protects it from insects. It's the same plant that produces anthocyanins that colour gin blue, and it's the same plant that's used in farming practices around the world as a fertility enhancing agent in agriculture for cattle. So it's a really wonderful plant, and we're in the process of doing the genome of it to understand just how many more cyclotides might be there, and it's very exciting to see that there's probably a lot more cyclotide to be farmed.
What's the, maybe you don't have to top of your head, but what was the sort of the radius of insecticidal activity where the plant was affecting the ones around it? Do you know?
Yeah, I think it was quite local. So it was just, this plant was actually introduced into Australia. It's actually present around the world, mainly in subtropical regions. I think originally coming from Indonesia and Madagascar, but so it's a plant that's been around for a long time. I think if it was brought into Australia as a forage crop for cattle, it's a part of the Fabaceae, or the legume family. So farmers were going to use it as a rotation in their fields. Being a legume, it could return nitrogen to the soil. So it was brought in as something that could be planted, off season to put nitrogen back into the soil and be a forage crop for cattle. And the cattle love it. We've also independently discovered that other cyclotide bearing plants can have these libido enhancing effects. So maybe the cattle have a reason for liking it. But we know it contains 70 odd cyclotides, and we know some of those cyclotides are actually active against nematodes, and we've actually tested them against nematodes of sheep and cattle. So it sort of might be that cyclotides have multiple roles. Natural insecticide for the plant, nematicide for killing worms in the guts of foraging cows and sheep, as well as potentially having aphrodisiac effects as well. So that's why we like cyclotides. They lots of potential applications.
Definitely. I'm smiling as big as I am because I'm thinking of like cows eating the crop and then pooping cyclotides all over the fields to regenerate like you said, the nitrogen content, but then also I don't know if the plants that grow within that field would then like still be like somewhat protected.
I think we should patent your idea. You've just introduced the idea of the circular economy from the circular peptides. So,
that's pretty cool. Well, the reason I was asking about the radius of the sort of the protection that's offered was, people will plant different things together in their garden, right? As like natural ways to get rid of pests. But if it's not that big of a radius, then they totally.
No, I don't. It might be that you could do that in a very local area, but it's more that often these days in crop protection, you don't necessarily have to kill an insect to protect your crop. You can change the behaviour of the insect. So I know, for example, with cyclotides, they are lethal at certain doses, and they kill insects, but they also stunt growth. So a larvae from a Heliothus moth, for example, will not get past first instar stage if it's eating a cyclotide containing diet. So there is that effect on growth retardation. There's the mortality effect, but we also see a behavioural effect where normally these moths will come and lay their eggs, and they'll lay a little bit here, a little bit here, spread it around a field, and then all the little larvae will grow and develop. But when they come across a cyclotide containing plant, or a plant that's been sprayed with cyclotides, they do what's called panic laying, and they don't like the environment for some reason. They lay all their eggs in one place, and these little caterpillars are cannibals. They eat each other, so it ends up, they hatch and then they all eat each other. So you end up with one caterpillar rather than 500. And the farmers don't mind that so much because it's reduced the
for one
so,
caterpillar.
it's sort of interesting, and this is something that is a little bit outside our expertise, but we're learning as we talk with farmers about how nature evolves things, not necessarily to kill something, but to change behaviours, and that can really have a big impact on the protection. And that's why it was just a chance observation that having a butterfly pea plant somewhere near a cotton crop, that it was noticed that the insects that normally would attack the cotton crops didn't attack the butterfly pea plant or any cotton plants, very, very close by. So observing nature is a good thing, Wendy.
Exactly. No, I think that's really cool. It's a good point too of trying to think, or I guess just through listening to each other and talking to each other, and like you said, not just in our fields but outside of it, and thinking of novel ways to interact. I was just talking to actually in episode 2 of season 2, Excitant Therapeutics. One of the co-founders in his laboratory, he does academics and he has this company, but in his academic laboratory, he's really interested in, can you overactivate certain elements of bacteria like proteins that actually leads to their demise. And he's thinking along the lines of like cardiac arrest, where you go into VFib and it's like it's going too fast. Can you do the same thing? And this is similar what you're talking about with the farmers, where it's like having a completely different perspective of what can be accomplished. So that's really cool.
Yeah. No, it's neat. And one of the pleasures of being a researcher but working in an applied area is you get to go out and talk to cotton farmers and find out what the problem is. And then learn new things and think about how we could, do interesting things with these molecules. So.
Yeah, for sure. Now, you don't only work with cyclotides. You do a lot of other somewhat disruptive things, some people might say, which I like personally.
No, look, well, pretty much everything we do is peptides. So I've become a complete peptide addict these days, and so we like venomous animals as well. Living in Australia, we have lots of spiders and snakes and cone shells, and a lot of our early work was in fact in conotoxins, where we take the venoms from marine cone snails. I might even have one here. I don't know if you've come across these, Wendy. This is a conus species. These are snails that live in the marine environment, bury themselves under the sand and have a little proboscis that they dangle out, and when a fish goes by, they harpoon the fish and inject a venom, and that venom can contain up to 1,000 peptides. And so we've been interested in, and often very disulfide rich peptides and post-translationally modified peptides. And Toto Oliveira in Utah is, of course, the godfather of this field, having really opened up the whole field. So we've had an interest for a long time in doing structures of these molecules and applying them in drug design examples. And in fact, trying to turn them into pseudocyclotides, because one of our colleagues from Australia, Bruce Livett, discovered one of those conotoxins years ago was analgesic, quite potent compared to gabapentin or morphine, but not orally active. But we were able to say it's a small peptide of 16 amino acids. We could add another 6 amino acids and cyclize it and turn it into a pseudocyclotide. And just doing that cyclization actually turned that into an orally active molecule in a rat model of pain. So we've been excited about being able to take a natural peptide from a venomous animal, re-engineer it to make it more stable and orally available, and pursue that as an analgesic. And down the track, we actually already done this. We've already put a gene for that molecule into plants, so that eventually we could produce this pain-killing conotoxin that comes from a marine snail into a plant where it could provide pain relief. And as particularly in the US, there's a huge opioid crisis at the moment, major problems with addiction and development of tolerance. These conotoxins act by a completely different mechanism and much less likely to lead to some of those problems. So,
Wow.
again, an example where sort of pottering around with nature and discovering things allows you to, and a little bit of peptide chemistry and peptide engineering allows you to sort of think of new ways of treating pain.
Right.
Which, as I'm getting older and getting sore backs, I think I might do my own clinical trials at some
There
stage.
you go. Yeah. Especially if you can eat it.
Yes.
Yeah, I agree. Again, I think that those examples in nature where we see really amazing molecules, and again, sorry to keep calling out this podcast, but the thing about this podcast is I get to interview really amazing people in a field that I love, because like you, I'm a peptide addict, and I got to talk to Helena Safavi, who was one of Baldemaro's students, and she and Danny Chow were studying those insulins from cone snails, where, I mean, that's phenomenal, right? Where it's like released outside into the ocean and is able to exert an effect. I mean, that's incredible.
Yeah. So. Well, yeah, no, look, it's amazing. And you don't think about these things. When you think about peptides, you think, how could they be working in the ocean? But we've got a project with a marine biologist from our university that studies the crown of thorns starfish, which is a major pest on the Great Barrier Reef, sort of one of the World Heritage areas of the world. And when you have an outbreak of these crown of thorns starfish, it can really devastate a local reef. And at the moment, the only way of dealing with it is they send divers out with a spear and spear the crown of thorns one by one and take them off. It's very laborious. The reef's 2,000 kilometres long, but Bernie Degman and Sandy, his collaborator, have discovered that that these crown of thorns secrete molecules into the ocean to attract other crowns of thorns during the spawning season. They've got all these interesting proteins, and so we've actually got a project with them to fragment those proteins and see if we can work out which are the bioactive signals. And the idea we've got, this is really crazy, is we could graft one of those sequences into a cyclotide framework and put it into a bait and have this in a trap on the reef that we could deploy. All the crown of thorns starfish see these signals coming out, thinking that it's party time. They all come towards the bait, and the door closes, and then we just remove it. So we've actually got funding from the Australian Research Council and one of the Great Barrier Reef authorities to try and explore this idea of using cyclotides as a stable bait, if you like, that we could attract pests and hopefully do something for the Great Barrier Reef. So that's sort of fun to be able to think of all these sorts of applications.
Totally. Yeah, that's really, at first I thought you were going to say just like putting cyclotides all over the ocean. I was like, ooh, slow down.
No. But the neat thing is, because these peptides are so stable,
you
can formulate them into all sorts of products. And there's a long way to go, and it's a very risky, high risk thing, but potentially a sort of an environmentally nice way of trying to deal with an environmental pest.
Yeah. Oh, that's amazing. Something else that I did come across when I was looking through your publications was some work that you've been doing with mirror image peptides. And there are a couple of things stood out. One was looking at what we've always considered peptide membrane interactions as achiral. And that's how we test to see if something's interacting with a protein or a receptor. Yeah. And you've uncovered some work there that looks like, no, probably not. That's not the case. Can you talk a little bit about that?
Yeah, sure. Well, look, that work was sort of built on the fact that as peptide chemists, we can actually make mirror image molecules. So we talked before about how we make these collider molecules. We just use solid phase synthesis, native chemical ligation. So we can do that perfectly well with D amino acids. And I guess my inspiration for that was from work that Steve Kent's been doing over many years, synthesizing D mini proteins for a whole range of reasons. So at some stage we thought that we'd like to make a D chelator molecule. We did it originally for fun, just to see if we could do it, but it turned out that there was two reasons for doing it in the end. If you make a D peptide and make a racemic mixture, then it turns out that it crystallizes magically well. All of our structures of cyclotides up until that point had been done by NMR. And being an NMR chemist, I trust them and believe them, but a lot of crystallographers were doubting, whether that cysteine knot could exist. But so we tried for years to crystallise chelata and other cyclotides. Our collaborator in Sweden got one tiny crystal once, and we did get a crystal structure using conventional methodology. But as soon as we made the D-chelata, made the racemic mixture, we had crystals within a week, solved the structure that afternoon. This was work by a wonderful postdoc in the lab, Conan Wang. So I became an addict of racemic crystallography, again inspired from Steve Kent's work. So I don't know why. It's something to do with the symmetry and the probability of more space groups being available. But we've now done that with a whole range of cyclic peptides, and it's pretty much worked every time. If you make a D peptide, mix it with your L peptide, you're pretty much guaranteed to get crystals. At least so far, that's, we've had 100% success rate. But then we thought we've got this D molecule, and we know that cyclotides interact with membranes, and everybody says, if there's a receptor involved, and you make a D peptide, there'll be zero activity. If it's a membrane involved, you'll have 100% activity. So we wanted to explore that, and with Sonia Enriques, who was a postdoc in the lab at the time, who's done all of our membrane binding studies, we wanted to look at a D cyclotide with a conventional membrane and then with the mirror image membrane. The problem is the mirror image membranes are very expensive to make. It was going to be $10,000 for a milligram or something, which we couldn't afford at the time. So we ended up developing the chemistry to make the mirror image membranes, and sure enough could find that D chelata with a D, effectively a D membrane, had 100% binding. L chelata with L membranes had 100%, but when we mixed them, we could see drops off, showing that membranes do have this chiral, there's obviously a chiral center in the head groups of the membranes we're looking at, but people tend to just brush that aside and think it's not important. But it is, it's chemistry, it's chirality, and it was neat to have that D probe molecule to show that.
Right. But something else. So the cyclotides do pass into the cells. We didn't really talk about that too much. So can you talk about the mechanism of how that?
Yeah. So one of the exciting frontiers in peptide science, I think, at the moment is getting to intracellular targets. So, everybody likes looking at cell surface receptors or extracellular enzymes, but the real action for oncology is, inside cells, protein-protein interactions. So we were always excited when we discovered early on that some cyclotides can actually get into cells. We still struggle with working out how much gets into cells, but we definitely have cases where we've engineered a cargo onto a cyclotide, see that it gets into a cell and has a biological readout that can only come from a cytosolic target. So we know that enough of them gets in. And we sort of know that there are multiple mechanisms, mainly sort of endocytic type, they go into endosomes first. So some cyclotides bind to membrane, most cyclotides bind to membranes, form, sort of get engulfed and formed into endosomes. Others don't actually bind to membranes, but get sort of engulfed by macropinocytosis, but still end up in endosomes inside the cell. So they're in a bag inside the cell, but they're not really in the cytosol. And so the big question is, how do they get out of endosomes? And I can't give you an answer at the moment, except stability, I think, is the issue, that if you're in an endosome and you survive long enough, eventually you leak out. The analogy I like to use is the door-to-door salesman. Someone comes knocking on your door trying to sell you something you don't want. You'll typically see them and shut the door and say no. Then they'll knock again and you'll open the door again and then shut it again. But if they keep on trying, eventually they might come and sell you that encyclopedia collection that you didn't really want anymore because you've got Google. So I think stability in a biological setting in endosomes is really important to get into the cytosol. There are now several examples where we know that cyclotides can get into cells, and that needs to be improved. So we're doing things like taking a cyclotide framework, putting a cargo on one loop, and then a cell penetrating sequence on another loop, so that we can enhance the uptake, or even recognize particular types of cells. So we'd love to be able to target a cyclotide to a particular type of cancer cell, so that we can be much more targeted and specific in our cancer therapeutics. And anybody who's been through chemotherapy would know that that's a very non-specific thing at the moment, and we need to have more targeted ways of treating cancer patients.
Even targeting towards like bacterial infections at specific sites. I mean, there's so much that you could do with that. One thing I will say about your comment about the endosome, I recently got to watch Jean Chmielewski give a lecture, and she was talking about targeting bacterial infections in different compartments inside cells, and what she was seeing was that you would first have a cell infected, and it'd be an infection that occurs in endosomes. And you would then put in the peptide, and that peptide would kill those bacteria, meaning there's some sort of exchange going on even after this. So it's, it's a good point to you, like that cells are dynamic systems, and if you have something stable enough that it will move around and
hang around long enough to have the effect. Which
yeah.
is, I mean, people say to us, cysteine not, does that survive inside the cell? And normally, the reducing environment, you might expect that you would lose those disulfide bonds, but that's not what we see in general, because the cysteine knot is so tight and reinforces itself. I think those cysteines survive, even a quite harsh environment inside cells.
Sure. So, have you done those sort of reducing environment studies just to show like when they start to?
Yeah, we've done it sort of qualitatively. I must admit, there's room for someone to come in and do that in a really qualitative way and understand, all the conditions, and that's something that's sort of on our books, but haven't quite got around to doing it. But just anecdoted in the lab, we do see that these are much more difficult to reduce than even your standard disulfide-rich peptide that doesn't have a knot. So I think it's to do with the fact that the three cysteine disulfide bonds are all right tightly interconnected with each other, and it's just hard for that reduction process to occur.
Definitely. I mean, that there's examples like human growth hormone. There's one disulfide that's super easy to reduce. It takes like minimal, amounts of reducing agent, and then the other disulfides are intact. So there's certainly precedent in the literature for that.
Yeah. The thing I guess we like about cyclotides, I mean, many people take a linear peptide and cyclize it by putting two cysteines at the ends and join it. But for me, that's a sitting duck cysteine ready to be reduced as soon as it gets into a suitable environment. Whereas we've got a head to tail cyclic backbone, so we've got a genuine peptide amide bond backbone across the whole molecule. And then the cysteine core is an independent moiety. So I think it's sort of the combination of both of those things that really makes cyclotides sort of the neutron stars of the protein world. They're super, super stable and really, really sort of tight and compact. And I should say, even today, we haven't come across another family of proteins that have both of those things, the cysteine knot and the cyclic backbone. That's a unique thing for cyclotides.
And then because you can have so much variability in each of those loops, I assume solubility can be addressed as well. So, because at first I was thinking fancy rock, it could be a fancy soluble rock. I mean,
we can, pretty much anything is fair game in a cyclotide, except for the cysteines. They're sort of immutable. They're part of the core, but pretty much all the other loops we can vary the amino acids at will. And we see, variations of 6 or 8 or 10 different amino acids at a particular spot in any of the natural cyclotides. The only other exception being there's a very strongly conserved glutamic acid in loop 1, and there's an absolutely conserved asparagine or aspartic acid in loop 6, which is the biosynthetic cyclization point. So there's a class of enzymes called asparaginal endopeptidases that recognize that asparagine, and they're natural proteases that every plant has, but some plants seem to have turned this protease into a ligase, which, if you think about it, first year chemistry tells us that reactions are reversible, so enzyme reactions are reversible as well. So proteases can be ligases. What we find is that those asparaginal endopeptidases in cyclotide producing plants have evolved, probably co-evolved with the cyclotides to really optimize that cyclization process. And we're now sort of excited about being able to recombinantly produce that class of enzymes and use them for biotechnology applications to cyclize a whole range of other proteins.
That's amazing. Of course, we ended up back with cyclotides as we were going from venoms through, but it's because there is so much. I think what you've uncovered is so much promise for those types of molecules. Well, we're at the end. Thank you for your time. Thanks for tuning in to this episode of Exploration Science. As always, we welcome your feedback and suggestions for topics that you'd like to see covered. You can leave those suggestions in the comments below or tag us on LinkedIn. You can also find this podcast available as a YouTube series by searching Exploration Science. Thanks again for tuning in.